脑人言。
那我简单的简，那我开始之前，我简单的介绍一下君达。君达呢是我师弟，他2015年到2018年是在南方科技大学读的本科，18到21年呢在 Wake Forest 的 Crystal 组，不是当时还不是 Crystal 组哈，在 Wake Forest 读研究生，然后随后呢就转到 Crystal 组，然后在2021到2024年呢到 Vanderbilt 大学呢。就攻读了博士学位。博士毕业之后呢，就去到了 C 的三的那个医学中心，然后从事人类电生理的研究。啊，那俊达你开始吧，我就不多说了。
Um, this talk will be in English, and but I can answer questions later in English and in Chinese. So thank you all for being here in the early morning on Sunday. And thanks a lot for the introduction and invitation for giving this talk of my latest publication. But it's actually the work that has been wrapped up like last year. Went through a pretty long review process and finally came up this year, last month actually. So the title I gave for today's talk is the adolescent brain structure, neural activity, and cognitive maturation. And we will only or mainly talk about the results from this publication shown on the right side on Nature Neuroscience, which I co-authored. And since Juan already introduced me, so this will just be a very brief introduction. I'm now a postdoctoral scientist in neurosurgery at Cedars-Sinai Medical Center in Los Angeles. I work with Dr. Uli Rutishauser, who's mainly working on human cognitive function and human intracranial phase. And before I work with Dr. Christos Konstantinidis and had a PhD in neuroscience from Vanderbilt University. And before that, it's in Southern University of Science and Technology in Shenzhen with a major in bioinformatics. So to start this discussion, I would like to, from this question, why study adolescent cognitive development? Or why adolescence is so important for cognitive development as a topic? I think the first reason is because adolescence is a period of rapid cognitive improvement. It's not like sensory or motor who is already developed after we were born, we can see pretty clearly with a full function. We can smell or we can touch things and feel things for the sensory part pretty clearly right after we were born. And I believe the motor and the coordination and even we can speak. I'm not talking about what's the content of the speech, but we can definitely control the muscle and speak well before the adolescence. But for a high level cognitive functions like decision making, like working memory, like memory in general, and response inhibition or value judgment, it's not so much until we were after adolescence. We reach the mature time period. So adolescence is a period of a rapid or dramatic cognitive improvement. Here, so what is the boundary or what is the definition of adolescence? In human, adolescence begins around 10 to 13 years old, which was the onset of the puberty, and it can last into the early 20s or up to 25. Here we see some of the human cognitive development studies from early literatures and our collaborators on the right side, who are also the co-authors of this paper. On the left, we see this performance of different individual, different type of visual spatial memory task, ranging as a function, ranging from 4 years old to up to 15 years old. And you can see this keep increasing in their task performance. And on the right, we are looking at some more precise measurement, like the accuracy of their memory-guided saccade and the latency of their memory-guided saccade, which is the eye movement in this working memory task as a function of age across 10 to up to 30 or above 30 years old. And we can see this dramatic change. So overall in human, we saw this improvement during the adolescence period. And with the development of imaging techniques, we are able to observe or measure the brain in the skull in a living person, not do the anatomy dissection. So people start to find out these parallel changes in the brain structure in anatomy as well. And here I show you this dynamic change or dynamic map of cortex thickness, where they show this human brain maturation parallels this functional development. And we can see this color represents the percentage of change in the volume of the gray matter. And we can see from 5 years old to up to 20, the brain was undergoing a rapid change in the gray matter and started with like all over the brain, and it matured earlier in the more posterior or occipital lobe, and eventually it's involved the frontal lobe, and for the prefrontal cortex, it's among the latest region that matured. Like up to 20, it's still changing. So, here comes the missing link of this, or when you apply for funding, the research gap, which is how do the changes in the different modality, the structure, the neural activity that we can record, and the behavior, where a lot of children developmental study or psychology study have found out interacts over this adolescence, which is a very critical period of cognitive development. And at the time of this study, surprisingly, we did not find any prior data set simultaneously measuring all the things I mentioned before, especially longitudinally. You record or you can test animal or human, but they come in, they do the test, and they are gone. They're not coming back for like every year or every month, and nobody tracks them for track the same person or same animal for over years. So we need to align this different modality, the different developmental trajectories across the different measurements. And before we jump to the content of this task, I want to give this like a last bit of background for the audience who are not that familiar with monkeys, especially for the developing monkeys. There are many reasons that the non-human primates, the monkeys, are popular for study brain functions, because they share many similarities in brain structure, in functions with humans, and they can perform many cognitive tasks, just like the ones that they use in humans for neurological check or just cognitive tests. And there's a lack of study on the neurophysiology of cognition in developing monkeys. There are definitely recordings on, like we call it normal monkeys or adult monkeys, but for young monkey or developing monkeys, not so much. So if you look at the time span of the rhesus macaques, the monkeys, they can live 15 to 20 years in the wild, and if it's in a controlled environment, like in the lab, you take good care of them, they can live up to 30 years old. And so there's a roughly 3 human year to 1 monkey year lifespan ratio. And this ratio is pretty linear, actually. It's not like dogs or cats that they have different portion of their life stage compared to humans. Monkey basically have the same like portion of different like childhood, the toddler stages, the adolescence, their aging. Like it's pretty linear projection to human life in this 3-to-1 ratio. And for a male rhesus monkey, they basically enter the puberty at about 3.5 years old, and reach this full sexual maturity at about 5, which roughly equivalent to humans' age, like 10 to 13 years old starting, and finish at 16 to 18 years old, respectively, which are also common seen as humans' adolescents. So it gave us a pretty good like model animal to study this development during this stage, because it's not too long that like human, you need to track them for years or tens of years, or too short, like they just snap and you miss the granulation in this temporal axis. So with this background, we started to design and create this project. We did this longitudinal cohort we designed for this project. It contains 8 animals. It contains 8 monkeys, 2 female, 6 males. And the way we do this longitudinally is to track each of them every about 3 months from their youngest age at 3 to up to 6.9 years. On average, the very first data point we collect on average on this 8 animals was 3.4 years old. And the very last one was 6.2 years old on average across 8 animals. So this longitudinal tracking just gave us more than 3-year multimodal data set, including their behavior, including their E-phase, and including the structural MRI and DTI, along with their physicals. So we need to track their actual physiology grooming. And by seeing their physicals, here's what we found first. We track this biometrics of all the subjects, including the morphological, their radiographic, and their hormone level over time. So we basically take them out and do the physical check every 3 or every 2.5 months and put them back. And we measure many things, like their body mass, how heavy they are, their femur length, which is their leg bone length, and their overall body length, and their canine length, which is the teeth they have. In the animals, it represents like they reach the sexual maturity. And for female monkey, we have their nipple size. For male monkey, we also test their testicle size, because these are the indicator of their sexual maturity, and they reach their real adulthood. And we also take X-rays of their distal radius and distal tibias, which is basically their forearms and their legs. So it's similar to the test a lot of parents will do to their kids, the bone age thing. Like they will go to the doctor and ask how many years left for their kids to grow taller. They basically are testing is the bone plate closed or not. So we're also testing that. And just similar, like the human study, or generally human observation, there are some kids reach maturity early, and there are some kids are lacking or like slow. Eventually they all reach to the adulthood, but there's a time difference. And we also see this time difference in over 8 animals. Here in this panel A, I show you like the number of growth plate closure as a function of their chronological age, which is based on their date of birth. Each of the line and dots, each color represents one animal. So you see 8 animals data here. So you see some animals, when they already have both their upper limb and lower limb bone plate closed, at the same age, some other animals haven't started yet, still like naive and like a baby boy. So we see this very profound individual difference, which means that we need a biological alignment. Just like human, when you measure the human adolescent development, you cannot just rely on when did they born, or when was their birthday or not. You also need to consider their puberty stages, their gender, which is a very big factor in this development, and also their demographic and all the background factors. So we also need to find a meaningful alignment here to align this data. Otherwise, we won't be able to make sense of this data set with such a large individual variability. And what we did is we defined a mid-adolescent age. So it's basically like a midpoint of the whole adolescent stage, which is defined by the time of both each monkey's both distal tibial epiphyseal closure, which is this point of each animal. So in total, we track 4 bone plate closure. And when they have 2 of them, usually the upper limbs closed, we call that this mid-adolescent age 0. And to make sure this alignment makes sense, or this is not some arbitrary or random thing we defined, we aligned the data again on the chronological age and on the defined middle adolescent age for each of the measurement. And here in panel B and C, it's the canine lens, which it's the longest teeth they have in monkeys. When they are babies, they have none. Like when they're still having a baby teeth, there's no canine. And for the adult animals, especially for the male, they have pretty long canine as their weapon. And on the B panel, we show this as a function of their chronological age, and we modeled this development or canine length. It shows a linear growth, which are not the thing we noticed or we observed in real life. Because in real life, the canine growth is like a eruption manner. So it has non-canine, so the length at 0, and suddenly, just a short period of time, the canine just grow to the full length and they reach adult and it's stable. The canine is not going to grow older when they are getting older in their whole lifespan. And using this mid-adolescent age actually capture this eruption manner. So very slow at the beginning, there's an eruption period of time, and then slow down to reach a top or reach asymptotic max. So this is how on this measurement, the mid-adolescent age alignment makes sense. We actually tested this alignment on all the measurement we did, including the body weight, the trunk length, the testicle volume, the testicle size, and the nipple length for the female. And we test them, or we align them, we model them on both the mid-adolescent age and their chronological age. And statistically, we test the model fitting goodness. And overall, we find out that using the adolescent age is superior, or it's better from a stats level, that it's a better model fitting. And to note, this mid-adolescent age alignment is blinded to the later or other measurements. It's solely based on their physiology or based on all these measurements we have. So in parallel of all the physicals we did to them, every 3 months or every 2.5 months, we tested their cognitive function development, use a battery of behavioral tasks. And in this paper, we only use the data from the working memory tasks as a benchmark of their cognitive functions or executive functions. I test this visual spatial working memory task with ocular motor delayed response task, the ODR task, which is a standard visual spatial working memory task. So basically, it's the subjects or the monkeys need to memorize this cue location. And after a delay period, they need to report where they think the cue was with the saccade, which is a quick eye movement. And on top of this ODR task, we have this ODR with distractor task, where there's a distractor happening during the delay period to distract them or interrupt this delay period working memory maintenance. And they still need to saccade to where the cue was. So first, at the very early time point, when they were only 3 years old, we find out that with a limited amount of training, the young monkeys are able to do it with almost 80% of correct, even at a very, very early time point across the 8 animals, which is a good thing. Like it makes sense. It means that the data we collect at the early stages are making sense. They understand the rule and they can perform it, or they're trying their best to perform it. It's not like when they're young and they totally don't get the task and we record just their random guessing or the non-milliness data. But it also leave a very limited room for improvement. Like it's already 80%. So they quickly reach a pretty good, like 95%, in 1 or 2 years. So we need a more precise and deeper look at their behavioral performance. So I hope you still remember at the beginning when I talk about the human study for this similar task, for the ODR task or memory guided saccade task, they measure the saccade accuracy or precision. And we are also using this similar metrics to measure it here. Here it's called the dispersion index, DI, which is basically measuring the area of the landing positions of the saccade to the same target. And in this panel D, I show you the real example from the data. It basically calculates the area of this, and you see this fitting ellipse. It's one standard deviation from their mean landing position. And just give you a sense of the degree. So this will be when the DI is 2.44. That's how clustered the saccades look like. And when it's 18.56, that's how clustered or scattered the saccades landing look like. So we track each session's average DI across this whole time period across all animals. And what we found is the largest improvement in DI or in the precision of their working memory occurred from -31 to 0 months relatively to the defined mid-adolescent age. And this shape also resembled or mirrored the human adolescent working memory studies I showed you before. So one way to perform this task better will be you hold your action, you think of it, you sit on it, you think of it, and then you saccade to make it correct. This sort of like speed accuracy trade-off happened in many cognitive tasks. So we are wondering if that's the case, if it's just an adult monkey or older monkey learned to sit on it for longer and to make a better report. So we tested another very important factor or metrics of the task performance, which is their latency, characterized as the reaction time of each trial. And we found out that the reaction time also got shorter. The reaction got faster over the same developmental window and assembled the similar trajectory. So there's no speed accuracy trade-off. Both the precision and the speed improved in older animals. And it's the same for both tasks. Upper two are in the ODR task, and the lower two are in the ODR distractor task. Although the task is harder, they are still following the same trajectory. And this trajectory also shares a common shape or development pattern as the developmental measures I showed you before, just for their physicals. So behaviorally, kind of verified that this mid-adolescent age alignment makes sense in this level as well. So let's pause here a little bit because people will start to argue that, okay, we know that it's a longitudinal cohort, so it's the same 8 animals 3 years later, and you has been training and testing and let them perform the task for so many times that they probably already been an expert on this, right? The adult monkey, the same adult monkey, he's been doing this for 3 more years than the young self. So they have to perform better. And I think that's the nature of the longitudinal cohort. And we need to make sure this is not because they practice and make it perfect. So on top of this longitudinal cohort, we also add a control. So now these 8 monkeys we call group A, as I introduced already. So we track them every 3 months from 3.4 to over 6 years old. And now we add another 4 animals as group B. They came the same age, like about 3, a little bit above 3, and they were trained and tested exactly the same as group A's first time point. But instead of coming out every 3 months, they just go back to their housing and do nothing. None of the tasks, not even other tasks. They just do nothing until they are over 6. And they come out to do the exactly same we did to the group A in their second time point. Controlled for their task exposure, controlled for the amount of training they have, and follow the exact same recording procedure to record their second time point. And we have another 2 monkeys serve as the group C. and they are just ordinary or normal adult monkey, monkey lab will buy when they're already over 6 years old. They're the young adults. They're already stabilized, and they are naive to any other tasks or training before. And we train them as a young monkeys at their first time point with the same procedure. And we compare the data from their first and second time point. On the left, I compare the first and second time point of group A and B, these two groups. So the only difference between the time point two between the groups are that the interval between the two groups, between the two time points in group B is much longer than this interval. And that accounts for the difference in their task performance between the second time point. And on the right, I compared the group A and group C's first time point. And the only difference here is like they were both naive before. And the difference is that group C came much older, 3 years older than group A. And that also counts for this performance difference. So we can confidently say that the training does not explain this improvement solely. The change actually reflects the development. It's really just because their brain develops. It's not because the adult monkey are doing much more. Like the adult monkey who are not doing much more are naturally gonna perform better than the young monkey, the naive young monkey at the beginning. So far I have showed you this developmental effect of working memory function during adolescence in the behavioral level. And there has been, like, this is a monkey study, so the perk is we can record. And there has been many imaging study, lesion study in human, and other monkey study have shown the role of PFC, the prefrontal cortex, in working memory. It's basically dominating or actually it has a pivotal role in the working memory function. So we recorded from PFC to see if this is the mechanism of the change in the behavioral aspect. On the left, this is a actual T1 image from one of the animal, showed you where we recorded. And you can see this is the principal sulcus, this is the arcusulcus, and this is you can see this is a cross section of where the recording chamber is. And this panel A is a reconstruction, 3D reconstruction of this monkey and the real location of where the recording chambers are. So this is the arcusulcus, this is the principal sulcus. We only recorded the anterior part of it, so only the area 8A and 46, we skipped this posterior frontal eye field to focus on the dorsolateral prefrontal cortex. In total, we recorded 2,131 neurons across all 8 animals. And all these neurons age, or like relatively recorded age when the recorded time when the of the monkey age has shown a like pretty well spread distribution in this panel B. So each dot is one section that we actually isolate the single neurons. So you can see it covers the pre, mid, and post-adolescence and continuously leave no gap. And the very first thing we have this single neuron data is to, I guess, look at the biology of the neurons, right? So we examine some of the firing metrics of the PFC neurons. First, we notice this is the fire rate. We test the fire rate and we find out that the fire rate in baseline, in the Q, when there's a visual stimulus, and in the delay activity, all increases along this maturation development, along this time frame. And it also resembles a kind of similar trajectory we already see a couple of times today, faster before the mid-adolescent point, and then saturated or plateaued afterwards. Another thing we look at this is the neural's variability. So, how variable or how stable they are firing. So for this part, we look at the mainly the baseline period where there's no real stimulus or task load or task demand. And we find out that the neural variability decreases, which can indicate a stabilization of the network of PFC neurons. So beyond this very, well, so now we have this and we want to see, is that correlate or is that just totally independent with the behavioral? So we did a very simple correlation between the trajectories to look at how similar, like we saw it with virals saying, yeah, it's pretty similar, the shape, but how similar? So we do this correlation of the different neurofiring metrics with their behavioral performance, their DI, and the reaction time. And here's just an example of the DI with baseline activity, the correlation, and the reaction time and baseline activity correlation. And it's strongly correlate, which can indicate that the firing improvements or refinement may reflect the mechanisms of their behavior maturation or improvement. So now, on top of this, like looking at the fire metrics in a stats manner, we want to see if it has any change when there is like a task demand. So we now look at this temporal dimensionality of single neurons during the task trial period. And we measure the effective dimensionality only in the temporal space. And we find out that the conclusion is the temporal dimensionality increase up to the mid-adolescence and then slow down of the increasing and start to become like non-significant in the age or maturation axis. And this high dimensional temporal activity may represent a richer time varying coding during this during the task demands. And to see if that is the case, we move one step forward to see the population level activity or dimensionality. So now we expand the single neuron temporal space to a full neuron space, which is constructed by temporal as one axis and units by condition as another axis. So we are now looking at the dimensionality in the population. And we find out this population dimensionality also shows this strong developmental rates before the mid-adolescent age. And it reaches its strongest just before this age 0 point. And the high dimensional population coding usually is modeled as a more flexible cognitive functions or improved cognitive flexibility, which can make the PFC to be better performing other or harder tasks. So there has been literatures or previous studies showing the mechanisms of PFC neurons and how it maintains the working memory information and manage the cognitive process and demand. It's basically by dynamically representing the different sensory stimuli in a task-specific manner. And that's why the PFC is a higher level region. It's not like the retina, LGN, or even V1 to V4. Like they won't treat the same stimuli based on their context. It is a square. It will just represent as square no matter what it means, no matter what it means. But for PFC, what it means or the task context matters. And we happen to have this distractor task to test this representation in the context dependent manner. And since behaviorally we find out that the monkeys are better doing this task, we want to know if this representation also changed or getting better. And the way we do it is to check the population representation of the visual stimuli. And the way we do it is, for example, in a whole population, in the whole population high dimension space, we can find each of the 4 Q, its location or its coordinates in that high dimensional space. And to visualize it, to make it simpler, I just denoise it to a 3D dimension. So each of this represent the population representation of each of the 4 Q. And among these 4 Q locations or Q representations, they can form a 2D plane here. And in the original high dimensional plane, the high dimensional space, they will form a hyperplane, or we call it subspace. And this will be the subspace we called the Q plane or Q subspace. And same for this same population of neurons during the distractor, there will be exactly same 4 visual stimulus serve as distractor, and you can also project them to this high dimensional space, and each dot will represent its coordinates or its representation. And the 4 of them will also form a hyperplane or subspace. We can call it the distractor plane. So imagine if this group of neurons, if this neuron population is not encoding the context, it is not encoding the context. They just simply represent where the Q is. It doesn't care about if it is a distractor or it is a Q. Then the dots should be overlapping if there's no noise. Like even there are random noise, the 2 planes should be like overlapping or very close to each other. And even you are saying that, okay, there's a temporal component there, right? This happened earlier, this happened later. This should be parallel, or their norm should be parallel, and their norms should also be parallel to the temporal axis. So it's just only move along the temporal axis. The overall shape or the overall orientation shouldn't change. But we find out it's not the case in the PFC. What we see is that the Q and distractor stimuli occupy a different subspace, like this 2 different planes, based on the context, because visually they are the same. When it's Q on the left, it will be the same square, same luminance, same shape, same duration even. But the only difference will be the monkey understand that this one is a Q. It's where it should be cut to. And this one will be the distractor. You need to ignore. You need to totally ignore and don't memorize it. Don't remain it in the memory. So now we have this measurement, and we know that they are representing or they are occupying different subspace. We can have a way to measure how different they are. How different are these 2 subspace? And the way we do it is to calculate this rotation angle between these 2 hyperplane, which also, actually it's calculated as an angle between the norms of each hyperplane. And we call this the rotation angle between them. And we find out and we construct different populations based on the time interval or age interval. And we resample, we subsample and resample to get the variation to calculate the rotation angle of each subpopulation of neurons. And the result is that we find out that this rotation angle reached its largest around 4 months before to the mid-adolescent age, and then decreased a little bit, and then is pretty stable after the mid-adolescent age, which we clustered as like late adolescent or young adult. So to put it in words, it will mean that the mature PFC create a more orthogonal representation, which is the population mechanism that can decrease or can prevent interference between the upcoming sensory input and the information in the memory, which is also what we see in the behavioral results, that these monkeys, when they grow older, they have this better resistance towards the distractor, and they can perform the ODR task and ODR distractor task better. So now jump to the third part of the main results. We already see this neuron changes in their firing rate, in the dimensionality, in the population firing pattern, and the encoding manner. And we also see this behavioral change as well. There are two type of, like, we want to dig one layer deeper to see the real biology or anatomy change, which can be the really physiology foundation of the changes. And in the field, there are two main structural brain change being frequently linked to the improvement in neural activity and computation. The first one is pruning, which basically means that you just for the infrequently used synapse, it will just die out. It will just, since it's not being activated, it will just disappear. So that's what called pruning. And another one is the melanization of the white matter tracks, which is modulated in a more activity-dependent fashion and has been proven can enhance the maintenance of working memory related information. So since we have the chance to do the imaging on this developing monkey, we wish to determine which aspect of the brain structure change that is the best to explain the changes in the neural activity as well as in the behavior. First, we look at this gray matter and white matter development trajectories globally. In panel A, shows you example slices of the monkey brain, and the color represents the percentage change in the different voxels or different subregions. This is an example, and B and C are the real data. B and C are different segmentation based on different atlas. And overall, we see globally the white matter volume raised strongly across the whole adolescent period. And the gray matter increased slightly here and then maintained pretty stable, if not decreasing, which is slightly different from the results we observed on human, because for human, gray matter peaked pretty early, like right after we were born, and there goes protracted pruning process. The gray matter will just decreasing, and when it reaches adolescence, basically it's very stable. And in the monkey, we find out the gray matter actually increased slightly during this period of time. And also, we also see this subcortical structure, which mainly the subcortical gray matter increased robustly. The pons, the thalamus, it all, the size also all increased. So to look at the cortex, we look at the 4 different lobes, and what we observed here, what we measured here is the first column is their volume of different lobes, and second is we break down to the average voxel thickness, which is kind of hustle to actually calculate this, and the surface area, which is again we want to see if there's more folding, if the sockets are getting deeper, etc. And different color represent the 4 different lobes. The red one was the frontal lobe. And in overall, we found out that the frontal lobe cortical thickness here showed a inverted U shape. So it increase and then decrease. And for the occipital and temporal, there's barely any change, which also makes sense because they are more posterior, and in human, we already show that they mature or they stabilize before the frontal. And for surface area and volume in general, it's pretty stable across all regions except the frontal. So it can verify that frontal is still the most actively changing one during this kind of late period after they were born. So we zoom in on the frontal, and now we break them to the sub areas where we break them into 4 different parts: the ACC, the lateral PFC, the motor cortex, and the OFC. And what we found out is that the lateral prefrontal cortex, the thickness of it peaked pre-adolescence and start to decrease after the mid-adolescent age, where the motor cortex also show a similar pattern. Here is the darker purple ones, the darker purple color. It represents the motor cortex. But for OFC and ACC, they are relatively stable across this whole time period we track. So moving from gray matter maturation and changes to white matter, the metrics or the method we use here is we use the DTI image we took from all these animals. And to quantify it, we use this FA value, the fractional anisotrophy. And we find out that for all the tracks we registered and identified, the FA value increases across most of the tracks, and specifically in this long range tracks showing, upper here. These are the long range tracks that go from anterior to posterior, or go from lobe to lobe, hemisphere to hemisphere, connecting and bridging the different lobes and brain areas. And for some shorter ones, like the projection and association fibers, they also show the strong developmental changes, but not as strong as these long-range projection fibers. And for the timing of it, this color represents the deeper or the darker the color, stronger the color is, means the more a change they have, and towards white it means like zero change or stabilize. You can also see this timing also resemble that fast at the beginning, then stable after the zero mid-adolescent age shape. And on top of the FA value, we also look at another or others DTI metrics, the RD value and the MD value. So these are just the raw figure from the paper. Each panel is one track. You don't need to go to the detail, but this will follow the F value, this is RD value, this is the MD value, and you can basically see the trend, which is most of the track we have seen the increase in the FA value and the decrease in the RD and MD value. And that is a strong sign of a progressive myelinization of that track bundle, that white matter bundle, axon bundle. And these results also resembled and agreed with human adolescent trajectories, where when they took image of human adolescents, like from kids to adults, they also see this very strong myelinization of their white matter tracks. And since it makes sense theoretically and also experimentally we observed, it means that they are a strong candidate. They can be strong candidates for expanding the improvement PFC dynamics as well as the behavioral improvement as well. And to see if this really correlates or if it can be a candidate of expanding the mechanisms, we first did a simple correlation analysis to see if the FA value can predict the behavioral performance, the DI and the RT, where here is a different tracks are FA value and their correlation to the DI. We find out that the DI strongly correlates with FA across multiple tracks. Same for the reaction time as well. It's not shown here if you go to see the supplementary figures. And we find out this most, and we on top of this correlation, we did factor analysis and find out that most FA values of the tracks have a higher prediction power than simply use the volume of different brain regions, especially from the prefrontal cortex, to predict their behavioral outcome. And at last, we do this multivariate analysis to consolidate and confirm our correlation to the factor analysis results, which turns out that the Y-matter tracks are a very strong candidate to predict the DI and RT. So that's very quick and summarize of the data. And the take-home message here will be, in this study, we found that behaviorally working memory precision and the speed improved across adolescence. And neurally, we find out that prefrontal cortex neurons firing increases, variability decreases, and in population, the dimensionality increases, which can be accounted for a better working memory performance. And we go one layer deeper to look at the real anatomy and biology and the structure of the adolescent brain. We find out that the Y-matter maturation is the best candidate to predict both the neural and the behavioral results during this time period. And altogether, they collectively characterize the cognitive maturation and the underlying physiological development. And I believe this dataset and this study provides the first longitudinal cross-model link in non-human primates. And for the implementations, this study provides the mechanistic basis of adolescent cognitive development, and it also highlights what's, I think, missing in a lot of these animal studies, that the importance of long-range connectivity maturation, which I think is actually increasing region of interest in the human research, because it's related to a lot of adolescent emergent psychiatry disorders like ADHD, like major depression, and specifically for schizophrenia. And on top of that, also for a lot of neurological disorder, like many types of epilepsy. And I mean, for today's talk, I only focused on the results for the scientific or neuroscience results on this. And I think another very important contribution or very important part of this study, of this longitudinal study, is that we adapted and leveraged a nonlinear modeling technique called GAMM, G-A-M-M, which is a generalized additive mixed effect model, which can better capture this non-parametric nonlinear effect of adolescent development, because you don't know what this shape will be. You see the scatters, you don't know what the shape will be. You cannot just assume, okay, it's a log or it is linear. So we use this nonlinear modeling techniques to capture this non-parametric nonlinear effect of adolescent development, not only on its typical imaging level, but also on multi-level, from their physical measurements to their behavior, to their neural firing, and to their gray matter and white matter metrics. And here's an acknowledgement. This is the list of authors of this of this paper, but there are more people, contribute to this project, for years as well. These are the trainees of Konstantin's lab at Vanderbilt, and also our many collaborators from multiple different institutes. I already graduated, so I cannot use Vanderbilt's logo and templates for this talk, but all the data curation and data analysis are done in Konstantin's lab at Vandy. And this 5-year-long project was funded by these 2 grants from NIMH. Thank you very much, and I'll take questions.